Hello and welcome to chapter 27. We are going to discuss the major development of a fetus and some hereditary traits and how those work. Um, so it's really easy to forget that the development of a human life is really a complicated and incredible event. This single fertilized oocyte growing into a complex individual made of trillions of cells, all from the combination of these two single cells. And that's what we're going to be looking at in this chapter. Uh, so you guys should have already covered the reproductive system. And we looked at the anatomy and the physiology of both the male and the female reproductive systems. And we described how gametes are formed. For males, those are the sperm. For females, those are the oocytes. Now we're going to talk about the developmental processes that occur when a fertilized oocyte develops into a new individual. So we're going to talk about the major stages of development all the way from fertilization to birth. We're going to look at the process of birth itself. And then we're going to look at and focus on what happens to both the mother and the newborn in the first few months after delivery. And we're going to talk at the very end, we're going to talk a little bit about how genetic characteristics are transmitted from parent to offspring, that mechanism we call heredity. So let's go ahead and get started. Now, human development is really going to begin at fertilization, right? When the two gametes unite. And it's going to continue all the way through life. Developmental biology is going to be the science that studies the changes in form and function that take place during these processes. You also have embryology, which is a subsection of developmental biology, and this studies the events during the prenatal period, that approximately 38 weeks of development inside the mother's uterus. It also includes a little bit, developmental biology also includes a little bit of the postnatal period, which includes from birth through all of the life stages, although technically postnatal may refer only to the short period following birth. So let's look closely at these periods in time. Now, the prenatal period is what people are usually going to refer to as pregnancy, and pregnancy embraces all of the events all the way from conception, when an oocyte is fertilized and begins to develop, until birth, and this includes all of the anatomical and physiological changes to the mother during this time. The growing offspring is generally referred to as the conceptus, and it develops during the gestation period of the pregnancy, which extends from the mother's last menstrual cycle until birth, approximately 40 weeks. And the gestation period is longer than the prenatal period because it includes that 2 weeks after the mother's last menstrual cycle, a date that the woman can usually recall. And we'll focus mostly on the 38 weeks of prenatal development, which is divided into 3 stages. The pre-embryonic period is those first 2 weeks after fertilization. If you guys remember, the secondary oocyte, and when that is fertilized by the sperm cell, the male gamete, this urges the secondary oocyte to undergo meiosis II, and it becomes an ovum. After that ovum is formed, the male and female nuclei fuse, becoming a single-celled zygote. During the pre-embryonic stage, the zygote is going to mitotically divide many times to produce a multicellular structure. This multicellular structure is a blastocyst. And the blastocyst implants or attaches to the endometrium of the uterus. Now, you also have the embryonic period, which extends from week 3 to week 8 of gestation. During this time, the conceptus is going to grow, fold, and it's going to form rudimentary organ systems. And you might expect, as the name gives it, during this time, the offspring is now called an embryo. The final stage is going to be the fetal period, and it lasts from week 9 to week 38, or until birth. In this stage, the conceptus is now called a fetus, and it grows larger and continues to develop until its organ systems can now self-function, or they can function without assistance from the mother. Once the offspring is born, it's going to become a newborn, and now those postnatal period of life begins. And here you can see the pre-embryonic period, the embryonic period, and the fetal period with their correlating conceptus structures. We have the blastocyst in the pre-embryonic period, the embryo in the embryonic period, and the fetus in the fetal period. And again, a small blurb there at the bottom about what happens in each event. Now, according to the CDC, some 1 in 8 couples can have trouble either initiating and creating a pregnancy or by maintaining it. Now, this can have issues on both sides. The male could be infertile due to a low sperm count. In female, it could be a variety of issues, hormonal problems with ovulation, ovarian disease, other uterine obstructions, things like that. And a common treatment for this is just going to be in directly manipulating the reproductive process using assisted reproductive technology, or ART. And now there are several types of ART. Some are going to be less invasive than others. One type, intrauterine insemination, also called artificial insemination. This is where a male's sperm is going to be collected, sort of washed and stripped, and concentrated to be mostly sperm cells. And so this is going to be placed directly into a woman's uterus at the time of her ovulation. These sperm are going to swim up the uterine tube and fertilize the oocyte. There is a similar procedure called gamete intrafallopian transfer, or GIFT. And with GIFT, the sperm and the oocytes are placed together in the woman's uterine tube. Now, fertilization is still going to have to take place normally and naturally, and the developmental steps all remain unaltered. Now, there are other processes that may involve external fertilization of the oocytes. Now, there's zygote intrafallopian transfer, ZIFT. Let's say a new zygote has been created in lab conditions, and now this zygote is now going to be put into the woman's uterine tube, where it will need to develop into a blastocyst and then implant into the uterine wall normally. There's also in vitro fertilization, which I'm sure many of you have heard of. And this is where oocytes are going to be incubated with sperm cells under lab conditions external to the body. And what's going to happen here is most of the steps of the pre-embryonic period are going to be allowed to complete. So by the time this conceptus is ready for implantation, it is at this point a blastocyst almost done with the pre-embryonic period. Now, it could even be an embryo at this point. And so the conceptus is now going to be transferred from the petri dish, as it's sometimes called, into the mother's uterus, where it has to implant naturally. And in some cases, these oocytes that are being manipulated are going to be directly injected either with a sperm cell itself, or what can happen is researchers or the lab technicians can strip a cell of its nucleus, a sperm cell of its nucleus, and they will inject just the nucleus into the oocyte. And that's what you see at the bottom of the image here, is the process of this in vitro fertilization, especially with injecting a sperm cell into an oocyte. Now, once the conceptus reaches the postnatal period, there are going to be rapid developmental changes that will take place, especially in those first few months, although they're not as dramatic as the prenatal period. The postnatal period is really broadly divided into 5 stages. There's the neonatal period, infancy, childhood, adolescence, and adulthood. Now, neonatal period is going to be from birth to 1 month, and we're going to look at this a little bit later. Infancy picks up immediately after that, from 1 month to 2 years. Childhood, from 2 years to the point of sexual maturation or puberty, when that begins. At this point, they begin adolescence, and that adolescence finishes when the individual is capable of sexual reproduction. Adulthood is what extends from the end of adolescence until death, and is the stage at which development ends and degeneration of tissues and organs, or senescence, begins. And of course, eventually senescence will lead to death, even without a specific disease stated. Now, some test yourself questions. The fetal period lasts during which of these periods of time? Is it weeks 1 through 2, 3 through 8, 9 through 38? Which weeks consist of the fetal period? It's weeks 9 through 38. That's when the conceptus was referred to as a fetus. Now, during the pre-embryonic period, the conceptus will divide into hundreds of cells via blank and become a or an blank, which will implant into the uterine wall. Now, the order here is important. So the division is what kind of division and what does the cell become? It divides by mitosis and becomes a blastocyst. Now let's talk about the pre-embryonic period and how it begins with fertilization, of a secondary oocyte by a sperm cell. And this period of time is going to continue until the blastocyst moves into the uterus and implants into the endometrium of the uterine wall. Now, during this period, some of these cells of the blastocyst are going to form extra embryonic membranes. That just means that they're outside of the embryo, and these work to support the developing conceptus. Now, if you guys remember from the A&P, or the reproductive chapter, we talked about the process of meiosis and how it produces gametes. Now, these gametes, or sex cells, are going to be resulting from they're going to be genetically unique haploid cells with half the number of chromosomes that are normally found in these body cells, right? Each gamete containing 23 chromosomes, and when the gametes combine, they restore the correct diploid number, 2n number, of 46 for the future cells of the new offspring. Now, the sperm cells that are ejaculated in this semen, and remember, semen is the mixture of sperm and other fluid secretions from the male reproductive glands. All of this semen is going to be containing chemicals that make the female reproductive tract more hospitable to this sperm and give it an opportunity to complete fertilization of the secondary oocyte. Each ejaculate tends to contain anywhere from 40 to 750 million sperm cells, but only a fraction of these ever actually reach the oocyte. And many of them are going to be killed by the acidic vaginal secretions. Some fail to penetrate the mucus of the cervical canal, or they can be, if they do penetrate that cervical mucus, they're destroyed by immune cells in the uterus. And sperm motility can be influenced by a woman's stage in her reproductive cycle. If she's in the secretory phase, the level of estrogen is high and cervical mucus is very thin and watery, which helps promote sperm motility. However, if the female is in the proliferative phase of her reproductive cycle, progesterone is high, and so the cervical mucus is very thick and viscous, and that can inhibit sperm motility. And many sperms survive these obstacles only to go up the wrong uterine tube. Remember, there are 2 uterine tubes, and only one of them is going to ovulate an oocyte at a time. In fact, by the time a sperm enters the female reproductive tract, no oocyte may be available at all. And an ovulated secondary oocyte is only viable for 24 hours after it's released from the ovary. And because they travel so slowly through the uterine tubes, they're usually in the distal end, called the ampulla, when they're fertilized. And pregnancy most frequently occurs when sperm enters the female reproductive tract during a 3-day window, from 2 days before ovulation to 1 day after, although sperm can remain viable for up to 5 days inside the female reproductive tract. Now, as I've said, fertilization is the fusion of the sperm cell and the secondary oocyte to form a zygote. Now, I want you guys to remember that this secondary oocyte that was released during ovulation is sort of frozen in metaphase 2 because it just doesn't continue meiosis 2 until it becomes fertilized. It also, once it becomes fertilized, it sheds that second polar body and now becomes what is known as an ovum. Now, both gametes, the sperm and the secondary oocyte, contribute equally to the chromosome count and the genetic makeup of this new conceptus. But functional roles are unique. For example, a sperm cell has to be very small and very motile because it has to travel such a long distance to reach that oocyte. In contrast, an oocyte is relatively large because it has to provide that cellular machinery necessary for that first week of development. And it is non-motile, meaning that it does not provide itself any sort of movement. Instead, it is simply swept along by cilia that line the uterine tubes and a sort of gentle peristaltic contraction that pushes this secondary oocyte from the ovary to the uterus. And in addition to these, we're going to have several events that need to happen between a sperm cell and an oocyte before fertilization can even occur. First is capacitation. Sperm have to undergo capacitation as they migrate. A sperm cell is going to move its whip-like flagellum, its tail, and that's what's going to make it move. There are some prostaglandins in semen that are thought to stimulate uterine contractions, which can help move the sperm further up the reproductive tract. And like I've said, although female orgasm is not required for fertilization, a female's orgasm can result in uterine contractions, and again, this may help in moving the sperm. And all of these processes together are called sperm migration. Now, before a sperm can actually fertilize an oocyte, it has to undergo capacitation. And this capacitation is a series of functional changes that make it fully motile and modify its plasma membrane so that it can fuse with the oocyte's plasma membrane. Capacitation is going to occur when these components of semen sort of weaken the sperm's plasma membrane, and these fluids in the female reproductive tract may also contribute to this process. And in addition, the oocyte itself will attract the sperm to its location using chemical signals. This is a process called chemotaxis. And what's interesting is that researchers have actually identified a receptor on human sperm cells that responds specifically to a chemical secreted by oocytes, and it appears to be progesterone. So these oocytes secrete progesterone, and the sperm sort of hone in on that. They're almost like bloodhounds tracking a rabbit or something. Now, an ovulated oocyte, a secondary oocyte, is going to be surrounded by that corona radiata that we talked about, those granulosa cells. And to reach the secondary oocyte, the sperm has to penetrate this cellular layer. And this is accomplished through sperm movements and an enzyme called hyaluronidase, which is released from the membrane-bound sac on the tip of the sperm called the acrosome. This hyaluronidase is going to catalyze reactions that break down the protein layer, and that serves as a sort of barrier between these granulosa cells. And immediately deep to the corona radiata is the zona pellucida. This zona pellucida is an extracellular matrix that is secreted by the oocyte, and it contains what are called sperm binding receptors. These receptors are an important site for communication between cells, and when a sperm binds to a sperm binding receptor in the zona pellucida, calcium ion channels in the sperm's plasma membrane open, allowing calcium to flow into the cell. This increases the intracellular ion calcium level, and it triggers the acrosomal reaction, which releases acrosomal enzymes, including additional hyaluronidase and acrosin. Acrosin is another protein, and these enzymes are going to catalyze reactions that sort of digest holes in this extracellular matrix, this zona pellucida. Now, numerous sperm cells are needed to release enough hyaluronidase and acrosin to break through the corona radiata and the zona pellucida. And that's why those males with low sperm counts, below 20 million, can have such an impaired ability producing offspring. Now, when the first sperm cell manages to go through the zona pellucida and bind to the oocyte's plasma membrane, immediate changes occur in the oocyte, and the zona pellucida occur so that no more sperm can enter. Only one sperm will ever enter the oocyte. And these specialized secretory vesicles are called cortical granules, and these are present in the oocyte, and they contain enzymes that destroy sperm binding receptors. When a sperm enters the oocyte, it triggers the release of these enzymes from the cortical granules. This is a process called the cortical reaction. So this cortical reaction prevents additional sperm cells from binding. On rare occasions, more than one sperm cell can enter the secondary oocyte. This is called polyspermy, and in this case, development does not proceed because the fertilized oocyte is now triploid, and these 3 sets of chromosomes are fatal to the zygote. Now, the sperm's nucleus is going to swell, and it's going to form what is called a male pronucleus. That's because the plasma membranes of the sperm and the oocyte fuse together, and with the fusion of these two nuclei, these two plasma membranes, what happens is the cytoplasmic contents of the sperm cell, including the nucleus, are going to be released into the cytoplasm of the oocyte. And this sperm nucleus now swells, and it's now called the male pronucleus. Now, at the same time, the fusion of the plasma membrane signals this secondary oocyte to complete meiosis II and become an ovum. The expelled polar body is gone, but sometimes remains visible at the edge of the ovum. This ovum's nucleus now swells and becomes the female pronucleus. Eventually, spindle fibers are going to form between the male and female pronuclei, and the pronuclear membranes break down, allowing these new chromosomes to now mingle. And if any part of this is ever interrupted, reproduction is not successful. It could be either because chromosomes are not allowed to intermingle, sperm cells never attach and become pronuclei. Any number of these processes can be interrupted, and reproduction will fail. And infertility, of course, is the inability for a pregnancy to develop after 1 year of unprotected sexual intercourse. And again, that can be either a problem on the male side or on the female side. So those sperm are going to undergo their capacitation as they migrate to the oocyte, and you see they're heading their way. They're following that secretion of progesterone, and eventually they will come in contact with the secondary oocyte, and they're going to begin doing the acrosomal reaction, and it's going to release all of those enzymes from the acrosome, this sort of green-looking tip. This it's going to release all those enzymes from the sperm cell. Now, if enough sperm cells manage to degrade through the corona radiata and the zona pellucida, what you're going to get is a sperm cell that manages to bind to these sperm binding receptors, and hopefully, what's going to happen? I'm sorry, these are the sperm binding receptors. These down here are the oocyte plasma membrane receptors. And so once a sperm comes in contact with those, the sperm is going to enter the oocyte, and it's going to stimulate this cortical reaction, triggering these cortical granules to sort of what essentially happens. It's not this isn't something I'll test you on, but what essentially happens is they destroy this zona pellucida, and what happens is water rushes in, and it creates a sort of fertilization membrane that pushes the corona radiata out so much further, and it completely rejects any additional sperm, so nothing can happen. No polyspermy can happen. And if you notice down here, it also destroys these plasma receptors, and it tends to destroy the sperm binding receptors. So there's nothing there. Even if a sperm does get into contact with the zona pellucida, there's no sperm binding recepting proteins. Now, once the sperm nucleus is injected into the cytoplasm, you see it's going to swell, and it's going to form a male pronucleus. So now we have a male pronucleus. At the same time, the oocyte is triggered to complete meiosis II and eject this polar body. And you can see in this image it is still attached. Normally, it may just be pushed out and secreted altogether. At the same time, the female nucleus, the ovum, the nucleus of the ovum, swells and forms a female pronucleus. Now, these two pronuclei join together and form this new what is essentially the new nucleus of the conceptus. And you see those spindles form, those spindle fibers form, and sort of allow these two pronuclei to come together, and the chromosomes will now intermingle. Now, the secondary oocyte has been fertilized, and the chromosomes from the male and female pronuclei are mixed in a process called amphimixis. And so now what we have is this diploid zygote. And approximately 30 hours after fertilization occurs, the zygote, the single-celled zygote, begins cleavage, a process of rapid mitotic divisions producing small genetically identical cells called blastomeres. And this division occurs too quickly for these cells to grow. And so for this reason, what happens is the cell number increases, but the cell size becomes progressively smaller. Each subsequent division takes less time, so that by the second day, there are 4 cells. By the end of the third day, there are 16 cells. And by the time you hit those 16 cells, the cells begin to differentiate and now are going to be the building blocks for all future tissues. At this point, the conceptus is known as a morula, and this morula remains covered by the zona pellucida. So here you can see some of those steps. Here you see the very beginning. This secondary oocyte is just getting out of metaphase. You see everything still lined up on that metaphase plate. Eventually, a sperm nucleus enters, and they swell and become the male and female pronuclei. And here we see the amphimixis, this combination of the chromosomes. That's the fertilization. So now, day 2, you have the cleavage forming. So now you have 2 cells, and these are blastomeres. And you can see just this progression of 2 becomes 4, 4 becomes 8, 8 becomes 16. And when it hits 16, it is now known as a morula. And all of this occurs in 3 days. Now, the entire time this is going on, all these mitotic divisions are taking place. The conceptus moves through the uterine tube toward the uterine cavity. Around day 4, you're going to see this glycogen-rich secretion from the endometrial glands called uterine milk. And uterine milk works to nourish the conceptus. And the conceptus is now called a blastocyst, hatching from the zona pellucida with these blastomeres reorganizing to surround what is now going to be an internal fluid-filled cavity. And by the time the blastocyst is ready for implantation, it has 2 distinct cell populations. It has large flattened cells as an outer layer, and these are called trophoblast cells. These surround the fluid-filled cavity, and then they have an inner cluster of rounded cells, the inner cell mass, or the embryoblasts. The trophoblast cells work to create the placenta, that temporary organ that provides nutrients and oxygen to the conceptus, and they also suppress the mother's immune system, working to prevent it from attacking the conceptus. The inner cell mass, a much smaller number of cells here, form the embryo proper, this actual developing body of the offspring. And here you can see the whole thing put together. You see as the oocyte is released here, let me get a better color here. As the oocyte is released, you see where all of these processes take place, where the sperm begins to attack in the ampulla of the uterine tube, where fertilization occurs here, and as we move forward, we get that morula as it just enters into the uterus proper. And at this point, day 4, we see the blastocyst now released from the zona pellucida, and you have these trophoblast cells and the inner cell mass here, with an inner cell mass here and the trophoblast here. And this is all what occurs at implantation. So once the trophoblast cells are formed, that the blastocyst is ready for implanting into the uterine wall. Now, during these early cell divisions, what can occur is the cells will separate into 2 groups, and they'll actually develop into 2 individuals. These are monozygotic twins, identical twins. They are 100% genetically identical and have a nearly identical appearance. They usually share the same placenta, and very rarely what can occur are identical triplets or even quadruplets. And this is all sort of I don't want to say a failure, but that's what it is a failure to normally separate during these early cell divisions. A more common type of twinning will occur from the ovulation of 2 oocytes at the same time. These are going to be fertilized by 2 separate sperm cells, and these are dizygotic or fraternal twins. They can be separate sexes, totally dissimilar in appearance, and multiple ovulations can actually result in triplets, quadruplets, or even greater numbers of offspring, but this is extremely rare except with the use of ART that artificially stimulates the release of multiple oocytes. Now, between 4 and 7 days after fertilization, that blastocyst that has formed is going to attach to the endometrium of the uterus, and this process is called implantation. Now, the trophoblast of the blastocyst is going to invade the stratum functionalis layer of the endometrium. It does this by secreting digestive enzymes that sort of degrade the endometrial lining. Now here the trophoblast cells are going to divide into two layers. On the inside there's a cytotrophoblast, otherwise called a cellular trophoblast, and here's where the cells are going to remain intact. Then you also have an outer syncytiotrophoblast where syncytiotrophoblast, where all of the cell's plasma membranes will disappear and their cytoplasmic contents merge. This really functionally makes the syncytiotrophoblast a large, single, multinucleated cell. Almost immediately after the syncytiotrophoblast is formed, it begins to secrete enzymes that digest the uterine cells. Interestingly, this is non-antigenic, which means that it does not trigger an immune reaction from the maternal tissue that it invades. The syncytiotrophoblast progressively invade the uterine lining over the next several days, reaching the maternal blood vessels on or before day 12, where it will surround the blood vessels, where it will surround these blood vessels and digest the walls of them. And what this does is it allows maternal blood to pool and fill cavities called lacunae, or intervillous spaces. The syncytiotrophoblast also makes contact with nutrients in the uterine glands, and by day 16, the blastocyst moves further into the lining, where it becomes covered by regrowth of those maternal epithelial cells on the surface. When the blastocyst is fully embedded in the uterine wall, covered by uterine epithelial cells, it is considered to be fully implanted. And here you can see some of these the processes that I was speaking of. You see between days 4 and 7, you have the blastocyst attaching to the stratum functionalis, and the trophoblast begins to differentiate into syncytiotrophoblast and cytotrophoblast. Day 8, you have the syncytiotrophoblast digesting the uterine wall and penetrating, getting further in. Day 12, you have some regrowth here of those epithelial cells, and you see lacunae begin to form. And by day 18, the cytoblast is fully implanted and covered by uterine tissue. Lacunae are really formed up now, and you see the blastocyst has begun to differentiate in cell layers. We'll talk about that here in just a second. Now, the syncytiotrophoblast is also going to be responsible for secreting HCG, human chorionic gonadotropin. And this stimulates that corpus luteum in the ovary to create hormones like estrogen and progesterone, and it helps promote placental development. The progesterone created manages to suppress menstruation by maintaining that uterus lining and prolonging the secretory phase. Some HCG is going to be secreted into urine, and that is where the basis for pregnancy tests comes from. HCG levels rise in the mother's blood until the end of the 2nd month of development, when a membrane called the chorion takes over the secretion of estrogen and progesterone. And at that point, HCG is no longer needed, and its level declines and remains low for the remainder of the pregnancy. Now, during the same period of implantation, while the trophoblast is becoming differentiated, the inner cell mass also separates and differentiates into 2 layers, the superior epiblast and the inferior hypoblast. Together they form a sort of flat bilaminar embryonic disc. It just means it is an embryonic disc that has two layers. Eventually, a small cavity is going to appear within the epiblast and become enlarged to become the amniotic cavity. This is going to surround the conceptus and fill with fluid. This bilaminar embryonic disc eventually differentiates into the three primary germ layers that eventually produce all of the body tissues. You have the ectoderm, the mesoderm, and the endoderm. And these three layers are really functionally the future blueprint for all organ development. Now, if for some reason the conceptus is not viable, if there's a chromosomal abnormality, it may die. And so this is called a spontaneous abortion or a miscarriage. And so if this occurs, the pregnancy will end. There are some rare occasions where the blastocyst implants into a site that is not the uterus, and if the pregnancy continues, the result is an ectopic pregnancy. This simply means a displaced pregnancy, and we'll talk about that here in the clinical A&P section. And so where last time we talked about the trophoblast differentiation, now let's look at the actual inner cell mass. You see it differentiates into the epiblast here and the hypoblast here, and it forms that sort of bilaminar disc. And again, it differentiates further, eventually becoming, you see this fluid-filled cavity, the amniotic cavity, being formed, and then eventually you see the endoderm coming in, the mesoderm, and the ectoderm all being formed. So now then, as I said, an ectopic pregnancy is when the conceptus implants in a location that is not the endometrium of the uterus. And this occurs in about 1 to 2% of pregnancies in the U.S. And almost all of these are going to be tubal pregnancies, meaning that they implant in the uterine tubes, but they can occur in other locations. Remember, there is a gap in between the ovary and the fimbri of the uterine tubes. So this oocyte could theoretically just wander into the abdominal cavity and it could implant there. You could also have some where they implant in the ovary or the cervix. And these ectopic pregnancies pose large risks to the mother, as the uterus is the only one that's able to expand, the only organ that's able to expand and sustain this growing conceptus. And if an ectopic pregnancy does not self-terminate in a spontaneous abortion, medical intervention is absolutely necessary to remove the conceptus. Sometimes it can be reimplanted into the uterus, but oftentimes what happens here, if it is untreated, this ectopic pregnancy will rupture, and it can cause serious bleeding, potentially even the death of the mother. Now, when they work together, the bilaminar disc and the trophoblast produce the extraembryonic membranes that first occur or appear during the second week of development, and they continue to development during the embryonic and fetal periods. Now, these membranes work to protect the conceptus and assist with vital functions like nutrition, gas exchange, and storage and removal of waste. Now, these extraembryonic membranes we're going to talk about are the yolk sac, the amnion, and the allantois, as well as the already discussed chorion. And we're going to look here at a figure that shows these membranes in their development. But first, let's talk about what they do. First, the yolk sac is what's going to arise from hypoblast cells and is actually the first of the extraembryonic membranes to develop. In some animals like birds, the yolk sac is the main nutritional source for the offspring, but in humans, cells from the yolk sac instead form part of the digestive tract, and they are the source of the first blood cells and blood vessels. And in addition, they're going to produce the first germ cells, and these germ cells are actually the prototypes or the precursors for the offspring's gametes that will eventually migrate into the developing ovaries and testes. Now, the amnion is going to be a transparent membrane that develops from the epiblast. Now, it grows to completely enclose the conceptus in a fluid-filled amniotic cavity only penetrated by the umbilical cord, whose vessels will connect the growing conceptus to the mother's blood supply. And the amnion is going to secrete amniotic fluid into this cavity. And this fluid protects the conceptus from any incoming trauma and helps maintain a constant temperature. This fluid also creates a freedom of movement that is important for symmetrical muscle development, and it prevents any body parts of the conceptus from drying out and adhering to one another during growth. At first, all fluid forms by filtering the mother's blood, but as the embryo's kidneys become functional toward the end of the 8th or 9th week, its own urine will contribute to the amniotic fluid volume. Then you have the allantois. The allantois is a small outpocketing of the posterior wall of the yolk sac, and this develops during the 3rd week. Now, the allantois has some early blood vessel formation, and these eventually become the umbilical vessels connecting the conceptus to the placenta. The tissue around the allantois becomes the connecting stalk, which forms the remainder of the umbilical cord. Inside the conceptus, there's going to be a portion of the allantois known as the allantoic duct, which runs from the umbilicus to the urinary bladder, which it actually drains. Now, the outermost layer is the chorion, and this develops from the cytotrophoblast and the syncytiotrophoblast, enclosing all of the other membranes and the conceptus. As the chorion develops, it forms outgrowths called chorionic villi, which are initially found around the entire surface of the chorion. But as the pregnancy progresses, villi grow only in the direction of the stratum functionalis layer of the endometrium and do not continue to surround the other membranes. By late in week 4 of development, those chorionic villi are blended with the stratum functionalis layer and begin to form the principal embryonic part of the placenta. There's a table here that shows a summary of these 4 extra embryonic layers, of their membranes and their functions. But first, let's actually look at some of these layers as development goes through. First, you have week 3. You have an allantois, a very primitive allantois here, with the future umbilical cord erupting from it. You see some of these chorionic villi. Of course, this will be coming from the chorion. The yolk sac tends to be pretty obvious. It's yellow, with the embryo proper being just this small bundle of cells here. And the blue sac here is the amnion, and the cavity that it creates is the amniotic cavity. Inside is the embryo. And as we progress into early of week 4, you see the yolk sac is beginning to distend itself, the allantois is elongating, and the future umbilical cords have some of those blood vessels that are formed and running from the embryo eventually out to the placenta. Running all through the chorion, you see the embryo is getting larger inside of the amniotic cavity, and of course this is the amnion still. Then by late in week 4, you see the embryo has really begun to differentiate and become a sort of almost recognizable shape. The allantois elongates further, and you see the vascularization from the allantois and from the umbilicus to this early placenta has really evolved. The yolk sac is still here. The embryo is still inside the amniotic cavity, inside the amnion, and the chorion you see has sort of thinned out and become just a very, very vascular structure. And here you have all those layers and their primary functions. So I'll give you a second to pause if you need to, but we're going to continue right now. Now let's do some test yourself questions. The blastocyst has two distinct cell populations. There's large flat cells called the blank and a cluster of round cells called the other blank. Now, which of these are the large flat cells and which of these is the cluster of round cells? The trophoblast are these large flat cells, and the inner cell mass is that cluster of round cells. Now, true or false, the allantois forms part of the digestive tract and is the first extraembryonic membrane to actually form. Is that true or false? That's actually false. That the first extraembryonic membrane to form is the yolk sac, and the yolk sac actually ended up forming part of the digestive tract. Now, at the end of the preembryonic period, the blastocyst has fully implanted, the inner cell mass is differentiated into the bilaminar embryonic disc, and the extraembryonic membranes have been developed. Now we can enter the embryonic period, which starts at the 3rd week of development, continuing through the 8th week. During this stage, the conceptus is referred to as an embryo. And this stage begins with a process called gastrulation. Gastrulation is the rearrangement and migration of these cells of the embryonic disc to form the trilaminar embryonic disc, meaning having 3 layers. So this is where we're going to get those 3 germ layers, all of them becoming major organ systems, in the process of organogenesis by the end of week 8. And now, although for the most part these organs are not functional until much later in development. During this embryonic period, the placenta finishes forming, and begins to provide that nutrition and oxygen to the embryo, as well as removing wastes. Now, in week 3, we start with a bilaminar embryonic disc containing 2 cell layers, the epi and hypoblasts. At this time, there's a thin groove on the dorsal surface of the epiblast called the primitive streak. This primitive streak actually elongates into the cephalic caudal line of the embryo, establishing the head and the tail regions. It also establishes the right and left sides and the dorsal and the ventral surfaces of this embryo. And this gastrulation is when this primitive streak has developed, gastrulation is what begins. It starts as some cells detach from the epiblast layer, moving into and then underneath the primitive streak in a process called ingression. The first cells that migrate this way becoming the inner germ layer. This is called the endoderm. And this actually replaces the hypoblast. After the endoderm has become established, the next cells that migrate, they migrate to a position between the epiblast and the endoderm, becoming a middle layer, the mesoderm. All of the remaining cells of the epiblast form the outer layer called the ectoderm. And by the end of week 3, all 3 primary germ layers have formed, making the embryo a trilaminar embryonic disc. And at this point, the ectoderm and the endoderm are tightly packed and surround the sort of loosely organized mesoderm. Now here you can see some of that ingression and this primitive streak being formed. You can see the primitive streak here and also here. And of course you do see this densely packed ectoderm, a densely packed endoderm, and a sort of loosely packed mesoderm. And this image here just shows those the ingression and the sort of shuffling of cells as they move to their eventual locations. Now, as you guys saw from that image, the trilaminar embryonic disc is pretty flat, and this has to change during the 4th week of development. 2 types of folding begin to occur, and they make this flat embryonic disc more cylindrical. First, you have some cephalocaudal folding occurring in the head and tail regions, and then you have some transverse or lateral folding, meaning the left and right sides curve and fold toward the midline. As sides move to closer together, you see the yolk sac is pinched off, leaving only a small area of its endoderm called the vitelline duct. As this cephalocaudal folding occurs, it creates the future head and buttock regions of the embryo, and the transverse folding actually creates the future trunk region and almost immediately forms the primitive gut, which later becomes the digestive tract inside the pelvic or the abdominal cavity. Now, in the process of organogenesis, those 3 primary germ layers differentiate into organs and organ systems. And when this occurs, or when it begins, rather, the primary germ layers have undergone those cephalocaudal and transverse foldings and are beginning to differentiate, with the embryo being approximately 15 millimeters long. When the embryonic period ends at the end of week 8, the embryo has almost doubled in length to be 25 millimeters long and will have very recognizable organ systems. Although they're not functional, not all of them are functional, some will begin to work. Now let's look at the major structures by examining the differentiation of each primary germ layer. So we're going to start with the superficial layers and end with the deepest. Now, the ectoderm, after the embryo undergoes cephalocaudal and transverse folding, the ectoderm is on the external surface of this cylindrical embryo. This arrangement is appropriate because the ectoderm forms the epidermis of the skin, and it is also responsible for forming a majority of the nervous system and many sense organs. In fact, the first major event of organogenesis is neurulation, which produces the brain and the spinal cord. You have some cells in the ectoderm that thicken and form a neural plate, which folds inward and deepens. The edges fuse and form what is called a neural tube, which will pinch off from the ectodermal layer, and the anterior end of this neural tube will become the brain. The remainder becomes the spinal cord. By the end of the 4th week, the anterior end of the neural tube begins to develop into those 3 primary brain vesicles, the forebrain, midbrain, and hindbrain. Now, between the layers of the ectoderm that form the epidermis and the neural tube, you have special cells called neural crest cells. These neural crest cells migrate throughout the embryo and give rise to various structures like cranial, spinal, and sympathetic ganglia, as well as the associated nerves. They're the pigment cells of the skin, the cells of the adrenal medulla, and connective tissue cells. Immediately deep to this primitive streak, you have some of those mesodermal cells at the midline, and they form a structure called the notochord. And the notochord serves to support and organize an embryo around a central axis. There are remnants of the notochord that will remain in the intervertebral discs as the notochord is surrounded by the vertebrae during development. Now, the mesoderm has on either side of the notochord, it forms these structures called somites, or block-like structures. And each somite has 3 regions. The sclerotome develops into vertebrae and ribs. The dermatome develops into the dermis layer of the skin. And the myotome develops into most of the skeletal muscles. And remember, this is all in the mesoderm. Now, on the lateral side of these somites are clusters of mesoderm called intermediate mesoderm, and there are sheets of lateral plate mesoderm. With the intermediate mesoderm, you form the gonads and the kidneys. With the lateral plate mesoderm, you develop the spleen, the adrenal cortex, and most of the cardiovascular system, the serous membranes of body cavities, and connective tissue components of the limbs. And with the endoderm, this is the internal epithelial layers of the digestive, respiratory, urinary, and reproductive systems. It also forms the cavity of the middle ear and that auditory tube. You have several glands and accessory organs, including the thyroid gland, parathyroid gland, thymus, parts of the palatine tonsils, and the majority of the liver, gallbladder, and pancreas, all springing from this primitive endoderm. And all body structures really derive from these three primary germ layers, and there is going to be a table here that will show you what comes from each layer. But by the end of this embryonic period, organogenesis has occurred, and although the organs are rudimentary. Now, there is an image here that we're going to look at some embryos, but at weeks 4 and week 8. I want you guys to note that at week 4, the embryo has a prominent forebrain and somites, while at week 8, the embryo looks almost human with different body portions. Now, during the embryonic period, the embryo is especially sensitive to teratogens, or substances that can cause birth defects or even death. There are some infectious agents that can produce a maternal illness like rubella that are teratogenic. Other teratogens include alcohol, nicotine, and many OTC or prescription drugs, like aspirin, anticoagulants, some antibiotics, especially Accutane, the acne medication. This results in severely life-threatening birth defects. Although possibly the most notorious teratogen is the sedative thalidomide. Thalidomide was prescribed to thousands of pregnant women in the 1960s to alleviate morning sickness, but an error in the chemical formula actually resulted in many infants being severely deformed with very short flipper-like arms and legs. Now here you can see those week 4 and week 8 embryos. On the left is week 4, on the right is week 8. These are not to scale. But on the left, you see it is very, very rudimentary. And you can see it does have a very prominent forebrain. Let me get a good color here. You see the forebrain here, and these somites all down the what would be the spinal cord. And then at week 8, this almost looks human with some very off proportions. So all of this development occurs in the embryonic period. And here you can see the body structures produced by these three primary germ layers. The ones that I've already talked about are going to be the important ones that I want you to know. A couple test yourself questions. What are the two types of embryonic folding? Embryonic folding. Cephalocaudal and lateral or transverse is also okay, as long as it's not translateral. You see, I threw that in there to try and trick you. Now, which two of these structures are created by the ectoderm? The ectoderm creates two of these, but none of the rest. The nervous system and the epidermis of the skin. Remember that. Now, the fetal period of development typically goes from week 9 until birth, which is usually somewhere around week 38. And by the 12th week, the placenta is fully functional and provides nutrition to the fetus for the rest of this gestation period. For this section, we're going to look at the development of both the placenta and the fetus during this period, and then we're going to finish with a big picture look at prenatal development. Now, during the pre-embryonic period, the developing conceptus receives nutrients from two sources: uterine milk secreted from those endometrial glands, and digested endometrial cells from the uterine wall. The conceptus is going to continue to receive nutrition from these digested endometrial cells until about week 12, when the placenta is fully formed. And the placenta is going to be the temporary organ that's shed after the infant is born, and this is the site of oxygen, nutrient, and waste exchange between the mother and the fetus. The process, in addition, the placenta is going to produce hormones to support the pregnancy. And the process of the formation of this disc-shaped placenta, which attaches to the uterine wall and to the embryo or the fetus through the umbilical cord, this process is called placentation. Placentation occurs during implantation, but most placental growth actually occurs during the fetal period. If the placenta grows in the wrong place, it can actually block the cervix of the mother's uterus, resulting in a potentially serious condition during birth, placenta previa, and we'll look at that here in just a second. And at week 12, you can see this sort of primitive or developing placenta surrounding the fetus at this time. We'll get more in depth on these structures as we move through this section. Now, in a mother with a case of placenta previa, all or part of the placenta will actually attach to the inferior portion of the uterus. It can be either near or on or covering the cervix. And most cases do result in live birth, but placenta previa can cause spontaneous abortion or even premature birth. This condition is also dangerous to the mother because as the uterus stretches to accommodate fetal and placental growth, the placenta can actually hemorrhage if it's attached to the cervix or become infected. And both of these can seriously increase the risk of maternal mortality. Typically, cesarean section, the surgical removal of the fetus, is the preferred method of delivery in cases of placenta previa. Now, the umbilical cord is what's going to connect the center of the placenta to the fetus's umbilicus, or belly button. And this cord normally contains two umbilical arteries that carry deoxygenated fetal blood away from the fetal heart to the placenta, and one umbilical vein that carries oxygen and nutrients toward the fetal heart. So these are kind of like systemic veins and arteries. These are kind of like pulmonary veins and arteries, where arteries go away from the heart, veins go toward the heart, and so these veins carry the oxygenated blood and the arteries carry the deoxygenated blood. Now, the umbilical cord also contains a soft connective tissue with a sort of jelly-like consistency. This is called Wharton's jelly. This insulates and protects these umbilical arteries and veins with its external surface only covered by the amniotic membrane. Now, if you guys remember that when the embryonic period begins, the blastocyst has already implanted in the uterus, and the chorionic villi have penetrated the uterine blood vessels. So those lacunae have been formed and filled with maternal blood. Eventually, these lacunae merge into a single blood-filled cavity called the placental sinus, and once placentation has begun, the stratum functionalis of the uterus is known as the decidua. So the region of the endometrium that lies beneath the fetus becomes the decidua basalis, and the region that surrounds the uterine cavity forms the decidua capsularis. And here you can see some of these structures. We see lacunae and this decidua basalis here. Decidua basalis here. We see one of the lacunae here. And we do see that it is all beginning to merge and become the placental sinus here. The placenta is actually going to be considered a unique organ because it develops from both fetal and maternal structures. the fetal portion formed by the chorionic villi, the maternal portion by the decidua basalis. And the chorionic villi contain those fetal blood vessels that are surrounded by maternal blood in the placental sinus, but maternal blood and fetal blood never mix because they're separated by the placental barrier. The barrier actually consists of maternal and fetal basal laminae, fetal connective tissue, the cytotrophoblast, and the syncytiotrophoblast. Now, some of these substances that are actually exchanged across placental barrier are going to be exchanged between fetal and maternal blood supplies. But again, the blood never touches. Since the maternal and fetal blood supplies are so close, oxygen and nutrients are able to diffuse from maternal blood to the fetus blood for transfer into tissues, and waste actually diffuses from the fetus into the maternal blood for removal. So let's look at how this blood flows through the placenta. You can see here that you see some maternal blood flowing through the decidua basalis and then through the maternal arteries into this placental sinus. Then the maternal blood touches the placental barrier, which allows this diffusion of substances, but not a passage of blood cells. Think of this placental barrier as a sort of screen that doesn't allow that only allows certain substances to get transfused, or diffused, like gases, nutrients, and wastes, because they're small enough to fit through the holes. Once diffusion has actually taken place, the maternal blood is now deoxygenated and carrying waste, and it flows into maternal veins and back into the mother's cardiovascular system. Fetal blood is actually going to leave the fetus and enter the two umbilical arteries, and then blood flows into the placenta and moves into fetal capillaries in the chorionic villi. These capillaries are on the fetal side of the placental barrier, where fetal blood then picks up oxygen and nutrients, delivering wastes, but again, no blood cells touch. Fetal hemoglobin does have a greater affinity for oxygen than maternal hemoglobin, so it picks up oxygen from the mother's blood at the placental barrier. Next, the oxygenated blood will leave the placenta and flow back to the fetus through the single umbilical vein. Now, if fetal blood and maternal blood were to mix, it would actually be very dangerous. For example, a mother could have blood type A and the fetus could have blood type B. They could actually both die because of the agglutination of erythrocytes caused by that incompatibility of blood types. And now, in addition to its nutritive functions, the placenta also functions as that endocrine organ, where the placenta assumes production of HCG, and as it grows, it results in the subsequent reduction of the syncytiotrophoblast. In addition, the corpus luteum will relinquish production of progesterone and estrogens, and placenta will take that role up. The placenta also produces human placental lactogen and placental prolactin, which help prepare the mammary glands for milk production. It also produces relaxin, which relaxes the body's muscles, joints, and ligaments, and presumably this works to facilitate stretching during delivery of the newborn. Now, during fetal development, the growth of the fetus is going to be characterized by the maturation of tissues and organs. In addition, fetal growth is rapid, and fetal size will change and increase dramatically. Limb or length measurements are typically taken crowned rump, CRL, crown rump length, or crown heel length, CHL. Now, an embryo that's 25 millimeters long can grow as large as 50 centimeters during the fetal period. Weight will increase as well, most obviously during the last 8 weeks, with the average weight of a full-term fetus ranging between 7 to 8 pounds. Now, a fetus and a newborn infant differ very noticeably in their body proportions. At the beginning of the fetal stage, the head is disproportionately large, with the lower limbs being very short. During the fetal period up to birth, the body proportions become more like those of an infant, and then after birth, the body proportions continue changing until adulthood, where the head is the smallest proportionally to the rest of the body. Now, there are some major events in fetal development. So let's look at these during the 6 months of the fetal period. In month 3, the fetal body lengthens as its head growth slows. So while upper limbs grow to their birth length, ossification begins in most bones. In the fetus's face, the eyes are well developed, but the eyelids are fused. The bridge of the nose develops, and the exterior ears are present but not prominent. During this period, the genitals are distinguishable as male or female, and then by the end of the 3rd month, the approximate length from crown to rump is 9 centimeters. During month 4, body growth is rapid. At lower limbs lengthen, joints form as the skeleton continues to ossify. Well-formed kidneys develop, the digestive glands form, and the heartbeat can be heard with a stethoscope. The fetus continues to develop, and it develops reflexes, and it will actually startle and turn away from loud noises and bright lights. Crown rump length at this point is typically 14 centimeters at the end of the 4th month. Month 5, growth will actually slow as lower limbs achieve their correct proportions. Hair grows on the head, and the skin is covered by this sort of downy hair called lanugo, and a whitish-grayish secretion composed of shed epithelial cells and sebum called vernix caseosa is also present. Both lanugo and vernix caseosa cover the skin, protecting it from this amniotic fluid. There is some brown adipose tissue that forms and helps with heat production, especially after the fetus is born. And typically, the mother can feel movements, a phenomenon referred to as quickening, as skeletal muscles contract. Crown rump length is approximately 19 centimeters by the end of the 5th month. Now, in month 6, the fetus gains significant weight. The eyebrows and eyelashes appear. Eyelids, which have normally been fused, are partially open, and the fetus's skin is wrinkled and translucent. And most importantly, the fetal lungs begin to produce their own surfactant, which is key to survival if the infant is premature. Crown rump length is typically 23 centimeters by the end of this month. Month 7, the eyelids open completely. Fat is deposited into subcutaneous tissue, so the skin is slightly smoother but still very wrinkled and very red. The fetus usually turns upside down, assuming the vertex position. In males, the testes begin to descend through the inguinal canal and into the scrotum, and then by the end of the 7th month, the approximate crown rump length is 28 centimeters. Months 8 and 9, these 2 final months, see fetal neurons beginning to develop their networks, organs grow and develop, and blood cells, especially in bone marrow, begin to form. The digestive and respiratory systems continue development, or complete development in the 9th month, with the skin being less wrinkled. Lanugo is also shed. In males, the testes complete their descent into the scrotum, and by the end of the 8th month, 30 centimeters, and by the end of the 9th month, 36 centimeters is the typical length. And here you can actually see some fiber optic views of fetuses at months 3 and 5. With month 3, you see very short limbs and a very large head. And at month 6, the limbs are much longer and the head is more close to postnatal proportion. Now, about 266 days after fertilization, a fetus is considered full term and ready to be born. The scalp usually has hair, the fingers and toes have well-developed nails, but if a fetus is born before that time, its tissues and organs may not have fully developed. We can talk about this in the clinical A&P section of prematurity. Now, all of the organ systems grow and develop after birth, but one system has to make rapid changes soon after birth to adjust to life without a placenta: the fetal cardiovascular system. The greatest anatomical changes between the prenatal and postnatal states occur in this system, with the fetus depending on the placenta and the umbilical cord for its blood supply. Cardiovascular structures seen only during prenatal development included the umbilical arteries, the umbilical vein, and the three circulatory shortcuts called vascular shunts: the ductus venosus, the foramen ovale, and the ductus arteriosus. And here you can see those being described here. Now, the foramen ovale is actually a hole in the interatrial septum that directly connects the left and right atria. The foramen ovale allows the blood to sort of skip ahead and avoid the pulmonary circulation by moving directly from the right side to the left side of the heart with systemic circulation. You have the ductus arteriosus as a short passageway that connects the pulmonary trunk to the aorta. This allows blood to move from the pulmonary trunk directly to the aorta. And we know that from our discussion of the placenta, umbilical arteries take fetal blood to the placenta for exchange with maternal blood, and then the umbilical vein returns that blood to the fetus. That blood first enters from the umbilical vein, traveling to the liver, but the immature liver cannot perform many of its filtering or storage functions. So complete perfusion is not really necessary before birth. So, for this reason, most blood from the umbilical vein bypasses the liver via the ductus venosus, which actually connects the inferior vena cava and flows directly to the right atrium of the heart. And after birth, these shunts and umbilical vessels close, and the regular circulatory pattern begins. Within a year, the flaps in the interatrial septum seal, the foramen ovale becomes a depression called the fossa ovalis, and in some cases, however, the flap does not really fuse shut, but remains closed anyway because of high pressure in the left atrium. Resistance and pressure changes in the pulmonary trunk and aorta cause the ductus arteriosus to close at about 3 months after birth, and the remnant of the ductus arteriosus is a sort of permanent cord called the ligamentum arteriosum. The ductus venosus similarly degenerates and becomes the fibrous ligamentum venosum, and the umbilical arteries become the medial umbilical ligaments, and the umbilical vein becomes the ligamentum teres, also called the round ligament of the liver. And here you can see some of those structures in the fetus versus the newborn cardiovascular system. You see how blood has to travel through the umbilical cord, and that completely bypasses many of the structures. And of course, those degenerate, becoming the ligamentum teres, or the round ligament. They become the ligamentum venosum. The fossa ovalis is what the foramen ovale becomes the fossa ovalis. The ductus arteriosus becomes the ligamentum arteriosum. All of these changes in the next few months after the offspring is born. And here you can see sort of the big picture. I'm not going to go over this at all, really. You guys, I've bloviated over it for an hour and a half. Fertilization to zygote, to blastocyst, to embryo, to fetus, to birth. You can see all of that development, and it talks about it here on the right. Now, I said I'd talk about prematurity, and an infant is considered premature if it's born more than 3 weeks before full term, so that's at about 35 weeks or earlier. And honestly, more than 12% of babies in the U.S. are born premature, And the truth is, the earlier the birth, the more complications that are likely. Most commonly, premature infants suffer from respiratory, digestive, and thermoregulatory difficulties. Now, the most common respiratory issue is infant respiratory distress syndrome, IRDS, and it develops in infants born before 24 weeks because surfactant is not produced in the alveoli of the lungs at this time. With no surfactant, the alveoli actually collapse during exhalation and become difficult to reinflate. Synthetic surfactants are what help premature infants breathe until their lungs develop sufficiently. You also have premature infants that face other difficulties like minimal sucking and swallowing reflexes, so they require feeding tubes. Underdeveloped livers can't produce enough clotting factors or albumin, so they bleed easily and develop swelling or edema very easily. Also, the hypothalamus is not fully developed, so they can't really maintain a constant body temperature and have to be kept warm in an incubator. You also have premature birth as the most common cause of infant death in the U.S. But because of the advances of neonatal care, survival rates are almost 30% for infants born at 23 weeks of pregnancy, and 50 to 60% at 24 weeks, 75% at 25 weeks, and more than 90% survive after 27 to 28 weeks. Do note, many infants who survive often face lifelong issues like cerebral palsy, learning disabilities, hearing or vision loss is also common. Let's do some test yourself questions, and then that's it. Now, the ductus arteriosus becomes what structure after birth? The ductus arteriosus becomes what structure after birth? It becomes the ligamentum arteriosum. Remember, ligamentum arteriosum. Now, the blank is responsible for secreting the hormone HCG, human chorionic gonadotropin. What structure is responsible for secreting HCG? The syncytial trophoblast is responsible for HCG production. All right, that's all we have for Chapter 27, Part 1. Check back. We'll talk about Chapter 27, Part 2, where we begin to talk about birth and all of the changes that occur in the mother during all of this fetal or conceptual development. All right, see you in the next one.